Yes, good evening, learners. Today we are basically going to have a lesson about carbohydrates and our learning objectives here that by the end of this lectures, at least you should have understood into details classifications and the structure of carbohydrates, the naming of monosaccharides, that's what you call the nomenclature. How do you name these monosaccharides? The aldose, the ketose, and all that. And according to the 5 carbon. We'll get into the details as we go through the lecture. And then the sources of carbohydrates, you also get to know that. And finally, you'll be able to relate what you'll be able to understand what glycemic index is and glycemic load, and also relate them to a diabetic patient. How can you apply that to that knowledge of glycemic index and glycemic load to a diabetic patient? Yes, now to kickstart with our lecture, it's an introduction here for carbohydrates. A carbohydrate is a biomolecule that is composed of carbon, hydrogen, and oxygen atoms. And the empirical formula is CH2 and O. With the N outside that N there is the number of repeated units. So we have different carbons, we have different carbohydrates. So then the N there determines which carbohydrate is that. So we'll get to understand later. And then carbohydrates are the most abundant in nature. They are more abundant than the proteins, they are more abundant than the lipids, and naturally they are the most abundant. So it is made, carbohydrates are made from plants through the process of photosynthesis. And then the examples of these carbohydrates include the sugars, the starches, the cellulose, and many more. The 60% of our foods in our diets are carbohydrates in most cases. So when you look at how people eat in a daily, in a meal in a day, you realize that someone has pork on the plate, someone has rice, and someone is having potatoes and have some beans or meat, and maybe a little bit of vegetable. So you find out that the biggest portion is actually carbohydrates. So in our diet, 65% of it is usually carbohydrates, and on digestion, it is converted to glucose, which is utilized by the body for energy. So we get to classify our carbohydrates. Carbohydrates are classified into simple sugars, and then oligosaccharides and polysaccharides. And the best classifications that we have actually here is the monosaccharides. Sorry about that, I had not finished the that's monosaccharides. So the classification here is monosaccharides, disaccharides, oligosaccharides, and polysaccharides. So monosaccharides and disaccharides are what we mostly refer to them as the simple sugars, and then the polysaccharides are the complex sugars, meaning they have more of that, including the oligosaccharides, and in most cases they are not digestible by the human body, the complex sugars. Though we will get to learn about them, and we find out that some of them can be broken down, like the glycogen. In a previous lecture we talked about it. They can also be broken down, though they are complex sugars. They are formed from the monosaccharides units that form together. I will get into the details later. And then monosaccharides here, starting with the first classifications, broadly, monosaccharides or simple sugars, they are the building blocks of complex carbohydrates. Now, you look at these building blocks, it means for you to form the bigger or the complex carbohydrates, like the starches, they are formed from the monosaccharides. It is joining of monosaccharides together. So that's when the two of them combine, they form what we call the disaccharides or double sugars, meaning two of them. We'll get to know examples like sucrose, lactose, which are also there later on. We'll get to discuss them. And if many of them combine, they form what we call the polysaccharides. And oligosaccharides is about 3 to 20 glucose units that have been, or monosaccharide units that have been bonded together. and they form glycosaccharides. Anything beyond 20, that's already a polysaccharide. So these monosaccharides include the glucose, fructose, and galactose. These are the major monosaccharides that is talked about everywhere, and I think they have been studied well into details. That's basically what almost everyone knows. Then monosaccharides are classified according to the number of carbon atoms. However, they can also be classified as aldoses and ketoses. Aldoses comes from what you call a functional group called the aldehyde, which I say pretty sure my cursor here is showing on where the ketone is and where the aldehyde is. The CHO, this is an aldehyde group. So if a simple sugar or a monosaccharide has this functional group on it, it becomes an aldose. And if it has the ketone functional group, and then it is ketose, meaning it is a ketone as well. So their classifications according to number of carbon atoms is right here on the left of the screen, on the left or on my left side here. You see the number of carbon atoms. Those with 4 carbon atoms are called tetrose, and the examples are right here: erythros and triose. And then the 5 carbon atoms are pentose. Hexoses are the ones with 6 carbon atoms. Heptoses are the ones with 7 carbon atoms. The list doesn't stop actually here. We still continue until 7, 8, 9, 10 octoses. There's octose, nanos. So it's a long list until 10. Then on the right side here, we are having the two kind of classification that you can see clearly that an aldehyde group is being indicated on the blue side on the image A. On the side A, this illustration here, the blue part shows the functional group and aldehyde. So this is an aldose, and we know if we have this is glyceraldehyde, we must have seen it somewhere during glycolysis. Then the ketone groups here is that it has a ketone functional group here, right here in the blue as well, and it is the dihydroxyacetone, which still we have seen the same biochemical process of glycolysis somewhere in the process. Then getting into the nomenclature, how do you name them? So one is that you add the prefix aldo to their names. One can define more closely a group of aldoses. For example, aldohexose. If something is a hexose, so you say aldohexose. Those are the 6 carbon monosaccharides. Aldopentose, the pentose here comes from the pentoses, like we discussed earlier. The pentoses are the ones with 5 carbon, the monosaccharides with 5 carbon. But if it has an aldehyde group attached to it, it becomes an aldopentose. Likewise, for the ketones, they also have the same, but instead of having the prefix, instead of putting the words in the beginning, as we do aldo in the beginning of this word, we put them at the end. So looking at some of the examples that we have here is that on the ketone side, those with 3 carbon atoms are called triose, and if they have an aldehyde group, we call them aldotriose. If they have a ketone group, they are called triolose. Either way you pronounce it, those are the spellings. And then we get into the one with the 4 carbon atoms, tetrose, and according to our naming here, nomenclature, you say those with an aldehyde group right here, you put the prefix aldo. So any tetrose goes with aldo and becomes aldotetrose. Likewise for tetrulose, which is a ketone with 4 carbon molecules. The same thing continues to the 5, 6, 7, 8, and 9 carbon atoms or monosaccharides. So I think with that we are so clear. And to start with some of these monosaccharides, one is a glucose. The glucose is the building blocks of carbohydrates. Mainly, most of these carbohydrates are built from glucose, and they are used in both plant and animal tissues. We know very well is the primary source of energy for all living cells. So it provides those cells which are including the brain, the nerves, the red blood cells, the lungs. All of this requires glucose almost extensively for energy. So meaning for your brain or nerve cells, red blood cells to function, or the lungs, including all other cells in the body, they really need this glucose to be available for use. However, the body can also divert to other sources if the glucose is deficient in the body, can divert into ketones and everything. That would be another discussion for ketone bodies and how they are used for energy in the body. However, for now, let us focus on this carbohydrates, which is a glucose, this monosaccharide glucose. It is the primary source of energy, and in the blood sugar is expressed. It is expressed as a milligrams per deciliters, and it can be also expressed in millimoles. For those who have ever seen some of these instruments used for measuring the levels of blood sugars, and some of them are in millimoles, some of them are in milligrams per deciliters. Some of them are in percentages depending on the type of blood sugar you're taking. The fasting blood sugar, the normal fasting blood sugar here is 70 to 100 milligrams per deciliter. What do I mean by a fasting blood sugar or blood glucose here? The fasting blood glucose is that levels that is measured when a patient have at least fasted for about 8 to 12 hours. So that is best. Most cases, if you're going to take your fasting glucose levels in the evening, you're told not to eat anything in the morning or in the night. So as a person, you'll be told to only have your supper and have your fasting blood sugar tested before you have a breakfast or any other meal in the morning. So that is what you call the fasting blood glucose. And its normal ranges are from 70 to 110 grams per deciliter. The carbon atoms are numbered from 1 to 6 in a clockwise direction, starting from the carbon immediately next to the oxygen atom. And we'll get into this because we have some illustrations during this lecture. We'll be able to see the numberings and where you start from. And this is the structure of glucose. We have two forms of glucose, which is the alpha D glucose and the beta D glucose. And on the right here is the alpha D glucose. You can see that on the difference is right on the first carbon where my cursor is. You can see in the red here is the numberings of the carbons. Like we said in the previous slide, that the carbon atoms are numbered. Carbon atoms are numbered 1 to 6 in a clockwise direction. Clockwise, the way the watch moves, the direction in which the watch ticks. So it is numbered in a clockwise direction, starting from the carbon immediately next to the oxygen atom. So we get here that this is the oxygen atom right here. When you are looking at this alpha D glucose, the oxygen atom is here, and then the first carbon is right here. So you continue counting from here. The next one will be 2, 3, 4, 5, and 6, and this is a hexose sugar. And so when you look at the next one here on our right side, the beta D, you find out on the first carbon atom number 1, there's also a difference in the position of the OH and H group. That's what makes this beta and alpha. So I guess you have seen the difference between the two. However, this is the linear form, which is, I think, in one of the questions which you should have discussed about the phasor projections, which is right the linear chain, and then this is the half of images of this or projections of glucose molecules, which is the cyclic form. So getting forward into what you call the fructose. Fructose is a monosaccharide that occurs with glucose in the sweet fruits and fruit juices. And fructose is also found in fruits, vegetables, and is present in invert sugars, a syrup made from sucrose and used extensively in the food industries. These are some of the sweeteners that manufacturing industries, the food manufacturing industries, use them for sweetening their foods. And it is formed along with glucose in the splitting of sucrose. Sucrose is the normal table sugar, the table sugar we have. That is what you call the sucrose, formed from fructose, and that is reacting with glucose. So once hydrolyzed, it gives you back the fructose and glucose. So it is formed along, fructose is formed along with glucose in the splitting of sucrose and is produced in the hydrolysis of various carbohydrates. So right here we are seeing is both the linear and the cyclic structure or image of fructose. Fructose looks is a 5-sided structure. It's not 6-sided like glucose and galactose. We can see now that on the numbering this time around there is no red, but this is the carbon number 1 right here where you're seeing my cursor on the cyclic structure on my right side or the right side of the screen. So the second right here is now on the ring, and then we continue right down, and then the 6th one is the one hanging right up there. So this is the numbering also on this. So you're supposed to be able to understand that these structures are quite different from each other. However, they are isomers, meaning they have the same molecular formula but different structural formula. That is what an isomer is. Hopefully you get to understand that. And then we get to galactose. Is a sugar that is rarely free in nature. In most cases you never find this in the free. It is found most in a bound together with other molecules, and it is found in the lactose, which is called the milk sugar. The milk that is the natural milk, the natural sugar that is found in milk is called the galactose. And possibly if you try to Google there and type in milk sugar, you'll be able to get something related to galactose. So it's broken down when lactose is broken down during the process of digestion. It will release for us what we call the galactose that the body can metabolize. Then going forward into the next classification of carbohydrates, is disaccharides. Disaccharides are molecules composed of 2 monosaccharide units. Just like you've seen on the right side here, we have a glucose molecule that is combining together with fructose to give us the sucrose molecule. And they're bonded together by what you call a glycosidic bond. This reaction is a condensation reaction, and it releases water molecules. Water is released from the formation of this glycosidic bond. That the OH group and the hydrogen on these molecules here interact and release hydrogen. We'll be able to see better pictures, I guess, as we proceed with the lecture. So this is what I said earlier on sucrose as the normal our table sugar that you know, the sugar you buy from the shops, which comes from sugar cane. So lactose is also another example, which is formed from glucose condensing with galactose, and this is what we call this is the sugar in milk, which I said earlier is called the milk sugar. Then there is maltose, which is formed from glucose, 2 glucose units that condenses together to form the maltose. And then we have third is the oligosaccharides, and oligosaccharides are part of the complex carbohydrates. These carbohydrates are low molecular weight polymers. They contain 3 to 20 sugar molecules, and because they are small, they are readily water soluble and often quite sweet. They are sweet because they are sweet and they are often used in some of these manufacturing industries as well as sweeteners. They are found in plant seeds, mainly legumes, which includes the beans, the peas, and the groundnuts. Yes, the groundnuts, and cannot be broken down by our endogenous enzymes, meaning the enzymes in your digestive system cannot break them down. So the non-digestible oligosaccharides are resistant to stomach acid. So meaning this acid itself cannot help in their hydrolysis, and also the action of amylase enzyme or intestinal hydrolytic enzymes cannot really help in breaking them down completely or to any extent. So what happens is that they enter into your large intestine intact, meaning undigested, just the way you ingested them or ate them. That's how they get into your large intestines, and they get fermented by indigenous bacteria. We have the bacteria that lives inside our digestive system. Particularly, let's talk about the colon or the large intestines. So this help in fermentation of these oligosaccharides, and they are fermented by this gut bacteria, and their ingestion is mostly accompanied by a complaints of gas and bloating. But why the gas and bloating is that during the process of breakdown, there is release of air, there is release of carbon dioxide in that process of the breakdown or fermentation. We all know, if you get to understand what fermentation is that during the process of fermentation, there's a release of that carbon dioxide or there's a release of gas. So that gas accumulation in the colon will result into the feeling that you have a gas inside you. And then bloating is a way that is a feeling of satiety. You feel so full and yet you ate nothing. And that is just because of the gas. And flatulence could also be accompanied by this. And flatulence here is a release of gas. Final summon is you go to the toilet and all what comes out is gas. No waste, no feces, and no any solid waste that you produce. Raffinose, which is one of the example of oligosaccharides, is found in sugar beets. It is a trisaccharide that is made from galactose, glucose, and fructose. So this is made of three monosaccharide units. This would be definitely easy to remember because you know the monosaccharides are galactose, glucose, and fructose. So when you combine them, all of them, and they condense in one unit, three of them, they form the raffinose. That is definitely easy to remember. That raffinose is formed from galactose, glucose, and fructose condensing together. That is three monosaccharide units. So we have stractose. Stractose is composed of two galactose molecules and one glucose, and then one fructose. Those are four monosaccharide units that have been condensed together. And this stractose, and raffinose, are found in the vegetables and the legumes. Then getting to polysaccharides, finally, these are large polymers of monosaccharides. They are formed by, sorry, there's a spelling error. They are formed by linking of many units of monosaccharides that are insoluble or form colloidal suspensions. What does this mean? These molecules cannot be dissolved, and so when something is not soluble, they form a colloidal suspension. And to understand colloidal suspension, let's look at someone that dissolves some solid that does not the someone that adds a solid that does not dissolve in water. When you stir it, you will see everything as if they're in a circular motion and moving. And you can try that practically with the sun. Just before they settle at the bottom of the glass or the cup, you'll be able to see them. After stirring, you'll be able to see them suspended in the liquid, and that is what we call a colloidal suspension. So about that, going ahead, we say the principal storage form of starch The principal storage form is starch or glycogen, and starch here is a polymer of alpha glucose and is a mixture of two different polysaccharides, amylose and amylopectin. So what are the difference between these two components or the molecules that make up starch is that we have an amylose, which is a long unbranched chain of glucose units, meaning there is no branch at all. So the glucose units just join themselves, that is by the 1,4 bonding, and there is no branching at all. Then we have that is branched, which is amylopectin, and there is also a form of branching in it. So that's basically how it is. Amylopectin is highly branched polymer of glucose units. So glucose units are formed and they are very well branched. They have branches. And glycogen, we definitely have a lecture about glycogen, which we had earlier. So we'll be able to refer there and get more details about the structure of glycogen and everything concerning it, including their metabolism. Then there's a structure of polysaccharides, taking into an example cellulose, which is one of the polysaccharides. And cellulose is the major component of plant cells. This is very important because in a later dates, we are going to discuss about fiber, and we'll be able to understand and appreciate cellulose, hemicellulose, and all those structures that are not digestible and the component of plant cell wall. And for this, let's first focus on this. Cellulose is a major component of the plant cell wall, and it is formed from beta glucose units, where each glucose molecule is inverted with respect to its neighbor. Meaning, if one is up the side, the other one it's more like upside down, just like the way you see here. For you to prove that, let's look at this diagram down. Sorry about that. We look at this diagram right here, that the first beta glucose and the second one, for you to realize that these are inverted, you look at the glycosidic bond. One is showing up, is projecting upward, then the second one, the second glycosidic bond, which is between the second and the third beta glucose molecule, is projecting downwards, and then the third one projects upward. That chain continues that way. This is just 4 beta glucose units. However, this chain is longer, so we cannot really have to draw a longer chain here, but to bring out the concept and understand that each of these units are inverted in respect to the neighbor. One beta glucose unit is inverted with respect to its neighbor. So if one faces the other side, the other is upside down. Of all, the other way to identify that this thing is upside down is that you look at the 6th carbon on each one of them, you realize that they are all inverted from the previous one. And finally, you end up forming what you call the cellulose, a complete structure of this kind. You can have an imagination of it looking on a brick wall that has been built and it has not been plastered, all the bricks exposed. You can really see that kind of joining, and that's exactly what cellulose looks like in its structure. So it has those parallel chains of beta glucose, and the spaces, the vertical spaces between the bricks are the hydrogen bondings, what keeps them together. And all these chains of beta glucose are held together by the glycosidic bonds. The alpha, it's the beta 1,4 glycosidic bonds. And then to bring all the different chains, the long chains of cellulose together, and long chains of this beta glucose that has been, all these chains, once they form a certain length of chains, they are joined together. And that joining is by the hydrogen bonding. So each one of them are parallel to each other. Each chain, a chain of glucose units, beta glucose, piled together in a parallel manner. So that's basically it about that. Continuing with the carbohydrates. They can be carbohydrates here can be attached by glycosidic bonds to non-carbohydrate structures, which includes the purines and pyrimidine bases, proteins, lipids, and aromatic rings as well. And so, looking into some of these examples. So, what do you call a carbohydrate that has been attached into a protein? That's a glycoprotein. You get, so those are the different kind of things that comes about. So, looking into some of the examples that we have right here is that if a group on the non-carbohydrate molecules to which the sugar is attached is an amino group, the resulting structure is an N-glycoside, and the bond is the N-glycosidic bond. And if the group is an OH group, which is a hydroxyl group, hydroxyl structure, it will be an O-glycoside, and the bond is an O-glycosidic bond. Now let's look at these structures here that we have a sugar here, which is the hexose, and resembles the glucoses and the galactose here. Bonding right here on asparagine, and on the upper asparagine chain here, you can see the side chain of this amino acid has an amino group. So on bonding with it, you have the N-glycosidic bond right here, and the resultant product is what we call the N-glycoside, a carbohydrate that is bonded to an amino acid that has an amino group. Then looking on the right side here is an amino acid called serine, which has an hydroxyl side group, and then when bonded to sugar, that reaction will give us what we call the O-glycosidic bond. So if you have a way of trying to understand this naming of this glycoside and their bonds, you just look at this, that if it is an amino group, it is N. Just on the bonding that you have, the bond is on nitrogen, that makes it an N-glycoside. And then the bond will be N-glycosidic bond. That's just for ability to remember what these structures look like. And then this likewise to the O-glycosidic bond, which have an oxygen atom here, bonding between the amino acid and the sugar. This makes it an O-glycoside, and the bond being O-glycosidic bond. Yes, then we get finally to what we call the glycemic index. After a meal, usually, the levels of your glucose in the blood rises to the maximum in about 30 minutes. That clearly indicates absorption and digestion taking place. Once you've eaten your food, let's say bread, let's say portion, or let's say you took some good fruit juice, or maybe you took tea that has sugar. The process of digestion definitely will take place, and the end product of carbohydrate digestion is the glucose. So this will be absorbed in your intestines or in your digestive tract, and then it returns, and then it will cause a rise after absorption into your blood. It will cause a rise in your blood levels up to its maximum. The blood levels would go so high, I can say it will shoot so high, and within about 30 minutes. And then later on, it returns slowly to the fasting level after 90 to 180 minutes. So, what does this imply? This means if your levels returns to normal, you have a normal system, or maybe this case is just for someone who's not diabetic, meaning the body is able to regulate everything and to bring back the blood sugars to normal within their possible and expected time. Then the height of the maximum and the rate of return to the normal vary with the nature of them. Some of this means make your blood levels to rise so quick. Some of these foods, for instance, when someone eats wholemeal bread, let's look at maize that has been grown and it has not been processed, or the bran has not been removed. and someone who ate maybe, let me say, bread that is refined, the two have a difference in the rate at which they cause your blood sugar to rise. Someone who ate the refined bread, the blood sugar will shoot very fast. Within the shortest time, the blood sugar is high. And then for them, someone who ate this whole means portion, which may not taste good to so many people because many people are not really comfortable with the wholemeal bread or foods that are having their brand inside without any other kind of processing. So that is actually very healthy, though it's not what people like. So we look in this that when someone eats that whole food, the rise in blood levels is not as fast as someone who ate processed one. So this here can be very important in application when you're going to look into things like handling issues of diabetes, to be very important. So at the end of this lecture, you'll be able to relate what glycemic index and glycemic load later on relates to a diabetic patient. So to continue with this explanation here, is that the height of maximum and the rate of return to normal vary with the nature of meal. So some meals are easily causing rise to your blood glucose levels. And this gives an indication of the rate at which the starch food are digested in the small intestine. So meaning some of these foods when you eat, they are easily digested so fast and they cause the rise in your blood sugars very fast. So we get to define glycemic index, we can define it as a physiological measurement which can be used to estimate the relative rates of glucose absorption from various foods. So this is just more like a measure. How fast, at what rate does this glucose get absorbed into your blood from a particular food? At what rate does glucose from bread get absorbed? And then maybe let's say also from portion, from some fruits and vegetables. And rarely vegetables have fruits. Vegetables have glucose, sorry about that. But fruits can have that glucose, and so I've not heard of a vegetable that has glucose yet. So we can major into talking about these fruits that has glucose in them. What fruit causes a fast rise to your blood glucose levels? So let's get into one of these examples here. If a subject ingests a portion of the test food containing 50-gram carbohydrates, and the blood glucose levels in the peripheral blood are measured every 30 minutes for 3 hours. Yeah. That is how you get to determine the glycemic index of someone. This what here you're seeing, the glycemic index is just an error. I guess it's supposed to be somewhere. The glycemic is supposed to be a continuous. Suppose I was trying to put in the glycemic load. So anyway, let's continue with the lecture. A glycemic index is a physiological measurement which can be used to estimate the relative rates of glucose absorption from various foods. A subject ingests a portion of test food containing 50 grams carbohydrates. Let's say we give you 50 grams of an apple. That's approximately a half. You get an apple, most of them are 100 grams. You just cut it in the middle, that would be about 50 grams. If you eat that, and then your blood glucose levels are measured every 30 minutes for 3 hours, we'll be able to know how fast your blood levels rise within a given time, within the 3 hours. Then we get into a concept called the glycemic load. This is also another tool, just like glycemic index, and they are so much related that the levels are still the same thing, glucose, and this is a more advanced and simple, and they say is a standard compared to glycemic index, and it measures how much a given serving of a food in grams causes a rise in your blood glucose levels. So this can be calculated by multiplying the glycemic index of a food by the amounts of serving in grams divided by 100. For example, if one ate 100 grams of apple, this is one whole apple, and has a glycemic, an apple has a glycemic index of approximately 38, then the glycemic load will be equals to 38 times 100 divided by 100. That will still bring you back to the 38 if you do the right calculation there. And this does not really require any serious calculation and everything like that. It's just a simple formula here. So this is basically how you determine what food can cause a rise in that. And how do we apply these two concepts in glycemic index and glycemic load? How do we apply them in our how do we apply them in a diabetic patient in management of these people as a nutritionist? This can be used in recommendation of the foods that a person should eat, having the knowledge of glycemic index. So you have possibly, if you have an option of refined bread and a wholemeal bread, you will definitely go for the wholemeal bread because it doesn't cause a high rise. It has a lower glycemic index compared to the processed one. So that's how you manipulate these things and try to give out the best for this say diabetic patient without necessarily telling them don't eat this and don't eat that. That has become our biggest challenge with the dietitians and nutritionists of the current days. Most of them, what they know, don't eat this, eat that, and everything, and that's it. So there's a comparison here: glycemic index versus glycemic load, and numerical ways, glycemic index, and numerical ways of showing how carbohydrates affect your blood sugar levels. So this is just in numbers, more like percentages. Say your blood sugar levels rises 38%, and that is what we call the glycemic index. Glycemic load here, it uses glycemic index, and is the amount of total carbohydrates per serving of a specific food to estimate how quickly a food causes blood sugar to rise and how much blood sugar levels will rise in total after eating. So you get to know that if I give this person an apple, his blood sugar levels will rise by this amount. If you know what the glycemic load of an apple or a food. So that is also very good in the management. That knowledge is also crucial in the management of all these diabetic patients. So the applications are right here. Knowledge of the glycemic index, GI, and glycemic load is important in management of patients with diabetes, and recommendation for the foods and the foods the diabetic patients should eat or moderate or avoid. What foods should you avoid? What should you moderate, or what can you consume freely without any problems? So having come to the end of this lecture, by now you should be able to answer the above questions now. What are carbohydrates? Those are biomolecules with the formula that C H2O into bracket N. You should be able to give us that formulas and then classify carbohydrates, which are the monosaccharides, disaccharides, oligosaccharides, and polysaccharides, and give us the detailed description of that. And describe the structures of the carbohydrates. We can now describe them, especially the cellulose and everything. Then you should be able to differentiate between glycemic index and glycemic load, and then you relate glycemic index and glycemic load to diabetes. So thank you very much, and we have come to the end of the slides. Wish you all the best. and thank you for having reached this.